So welcome everybody to the keynote session on food security in an unsecured world. So I'm very honored to co-chair this session together with Lise KORSTEN from the University of Pretoria and Lon Vugbald from Agriculture, Agrifood Canada. My name is Giuseppe Stancanelli from European Food Safety Authority, Italy. So the session will start now and will last until 6 o'clock. We have 3 speakers. Each speaker will give a keynote speech for 20 minutes, and then we'll have 5 minutes for question and answer from the public, and you can do through the app or come down and talk to the microphone. So now I let the floor to Lon.
My name is Lone BUCHWALDT. I'm a plant pathologist with Agriculture and Agrifood Canada in Saskatoon, Saskatchewan, which is on the prairies in the middle of Canada. It is my honor to introduce the Glenn Anderson Lecture as part of this session on food security in an unsecure future. Here you see a portrait of Glenn Anderson, a Canadian scientist. He was born in 1924. I have borrowed some excerpts from the first lecture honoring him. It was given by Norman Borlaug in 1990. And also I used an editorial by Werner Higgins, who was a contemporary plant pathologist at the University of Toronto. So as a young boy growing up on a farm in Ontario, Canada, Glenn experienced firsthand the economic harm to human welfare caused by the Great Depression in the 1930s. It coincided with drought and topsoil erosion on the Great Plains of North America, which devastated crops and livestock production. Glenn Anderson studied many aspects of crop protection and graduated with a PhD from the University of Saskatchewan. Initially, he worked on wheat breeding and resistance to rust diseases for the Canadian federal government in Winnipeg, Manitoba. In 1964, Anderson joined the All-Indian Wheat Improvement Program supported by the U.S. Rockefeller Foundation and assumed the scientific and de facto leadership. He created groups of interdisciplinary and talented people who managed a large network of test sites and developed crop production packages using new technologies, including dwarf wheat varieties initially developed in CIMMYT, Mexico. Glenn Anderson traveled extensively and constantly worked to convince governments and policymakers to provide new affordable technologies that would benefit farmers. During Anderson's 8 years in India, wheat production almost tripled and the country became self-sufficient. This success became known as the Green Revolution. Similar success was achieved in neighboring countries, Pakistan and Bangladesh. Glenn Anderson joined CIMMYT in 1971, first as an associate director of the International Wheat Program, and in 1979 he became the director after the retirement of Norman Borlaug. Borlaug described Glenn Anderson as a man of unusual intellectual capacity, who had acquired an excellent education across many disciplines. Glenn was a natural leader, an excellent teacher, skilled at identifying young students with potential. Some of CIMMYT's best wheat scientists at the time were identified by him. The International Wheat Program was extremely productive and successful. Anderson was instrumental in establishing a collaborative research program with CIMMYT and several research institutes in Brazil. As a result, new wheat varieties out-yielded the old ones by 50 to 100%. Glenn also supported CIMMYT's Triticale improvement program, which became an established crop in Poland, Portugal, Spain, Tunisia, and Australia. Anderson was the main author of several book chapters in Wheat in the Third World, published in 1982. One chapter focused on India, a country he regarded as his second home, Bangladesh, a country he greatly admired and to which he gave much thought, and a chapter on future research, a favorite topic for Anderson, the planner. Sadly, Glenn Anderson fell ill during a research visit to Africa in 1981. He was quickly reunited with his wife and 5 children in Winnipeg, where he passed away. A lectureship award was created by the Canadian and American Phytopathological Societies to honor the accomplishments of Glenn Anderson. This award sponsors an invited lecture at the International Congress of Plant Pathology or other joint meetings between our 2 societies. The award recognizes the contribution of an outstanding, internationally recognized plant scientist working to secure the world's food supply. The lecture is subsequently published in the Canadian Journal of Plant Pathology. It is my honor to introduce the 9th Glenn Anderson Lectureship to Dr. Bram GOVAERTS, who is the Director General of the International Maize and Wheat Improvement Center in Mexico. GOVAERTS graduated with a PhD from the Leuven University in Belgium. Dr. GOVAERTS bring together multidisciplinary science and development teams in order to integrate sustainable, multi-stakeholder and sector strategies that will generate innovation and change in agri-food systems. Together, they seek to combine the right seed and conservation agriculture production practices while also integrating farmer knowledge. His work will transform subsistence agriculture and failed farming system into productive and sustainable production units in Africa, Asia, and Latin America. Among other awards, GOVAERTS have received the Development Cooperation Prize from the Belgian federal government, the Norman Borlaug Award for Field Research and Application, the Premio Techno Agro Award from farmer organizations in Mexico, and he is also a fellow of the American Society of Agronomy. And with that, I will introduce, I forgot to change. Sorry, but it was pretty much what I said. I just forgot to change the slides. But we will start. GOVAERTS, are you close by? Thank you. You're welcome.
So first and foremost, thank you so much for all being here. I know it has been already a very long day. And second, I'm humbled by receiving this recognition, and I want to thank the 3 chairs. I think this is my first lecture ever where I have such a distinguished 3-chair that is presiding the panel. So thanks a lot. I do want to highlight that I received this award in name of multiple colleagues in CIMMYT and also our partners throughout. I also have to make a disclaimer. I am not a plant phytopathologist, I'm just a simple agronomist. So what you do when you have to give a lecture to smart people in the room, you call on other smart people to help you to put the lecture together. So I want to recognize my co-authors from CIMMYT that have supported this lecture. First and foremost, let's never forget that we are standing on the shoulders of giants, and I think for sure Glenn Anderson was one of those giants, and you have heard the introduction. Just one maybe other element, within CIMMYT he was very well known for being the father of thinking about multi-location yield and pathology testing sites throughout the world. He was also a firm supporter of the training and capacity building of young scientists and the new generation. And I will, of course, get back to that during the lecture. A bit of the history of CIMMYT, the organization that I have the honor to lead today. It started all with the dwarf wheat potential, or the new wheats that were generated by Norman Borlaug, and you see him here in this picture standing next to Henry Wallace, also known as the father of hybrid maize, back then vice president of the US. And together they convinced the Rockefeller Foundation to invest and to generate this program of special studies to send those new wheat varieties to India. And it was Glenn Anderson's task to convince local actors, local farmers, local decision makers to use those varieties, and through that generate what we would call today impact pathways. And as many of you know, it was this effort that actually generated the basis for the Nobel Peace Prize that Borlaug received in the 1970s for having saved 1 billion lives. It's these examples that we still take in mind today in CIMMYT to keep generating benefit for farmers globally, and it is summarized in a yearly benefit of $3.5 to $4 billion annually. Before you think we're very rich, those $1 billion go to the farmers. They don't come back to CIMMYT directly. Luckily, it is others that benefit from that. 70% of the wheat and 50% of the maize is attributed to some of the efforts. But when I talk about CIMMYT, I don't talk about the organization per se. I talk about several of you that through your efforts are collaborating with us, and I want to thank you for what you do every day. When discussing with decision makers, and especially ministers of finance in multiple of the countries that support us, they want to hear return on investment. Well, for every dollar invested in the wheat research, we have between $70 to $100 of return. And in the maize research, it's between $22 and $35. So next time you need to convince one of your colleagues, you can use these numbers. We are active in over 50 countries. Now, this was the history. And as my grandfather would say, if you want to know your future, learn about your history. We have reviewed that history, and if we now fast forward to today, I want to highlight that we have a huge challenge ahead of us. And I call it the challenge of the 4 Cs. We're still looking at post-COVID. An experience that has learned us that the resilience of our agri-food systems was different than we probably had expected. I don't have to explain you the second C, climate change. Just go outside of the door and you know what climate change means. If you would have told me 3 years ago that only 2,500 kilometers from lovely Lyon there would be a war going on, I wouldn't have believed you. Today, we have a conflict in between Ukraine and Russia that is affecting our global food supply. And of course, the cost of living is hammering hard on the livelihoods, especially of those that are already very vulnerable in Latin America, Africa, and South Asia. It is that challenge that all of you have the responsibility to take into account when you work every day. Because 40% of the world's food comes from smallholder farmers, and 50% of the people going hungry worldwide work on smallholder farmers. It are also those that their food security and livelihood trajectories depend very strongly on plant health management, and let that be what you're contributing to. And you cannot see that in an isolated manner. Please see all your work within the global agri-food system that starts from the biodiversity and ends by consuming a healthy, affordable diet, hopefully sufficient so they have what they need to live a healthy life. And in the middle, we generate seeds, we generate agronomic systems, we generate markets, local, regional, and global. And of course, there's a whole processing element to that. Go even to a broader system. It was when Lloyds of London asked us, can we actually map what food security means, and does that have an influence on what we are, the biggest reinsurer globally? And in order to show that, we took a couple of events that have happened in the past 20 years. And we said, what happens if you put those events together in 1 year? What would be the impact? And the events we chose were El Niño, warmer temperatures, and UG99. I was told very emphatically by one of the chairs, don't explain the whole UG99. All of you know about that, and for sure, if you want to know more, there is Dave Hudson here in the room, and several of you that can give a special session about that. But still, if you take those 3 events together, and instead of them happening in 1 year, you put them at the same time in 1 year, the impact on the global food system was the following. Wheat prices would go up with global production losses of wheat would go up by 7%, maize 10%, soybean 11%, and rice 7%. Of course, this has to do with substitution between those crops. Price increases would be by 4 and by 5 for rice, and human cost would be humanitarian crisis, food riots, but more importantly for Lloyd's of London, 10% of the stock impact in the EU and 5% in the US. This is the importance of your work. It is sitting and contributing in the middle of what is global stability. And in order to reflect that, CIMMYT worked on a 2100 vision. And when I stepped forward for the first time to the colleagues in CIMMYT and said, we're going to look at the 2100 vision, for sure the first reaction was, now that is very far away. For those of you that have small kids, small nephews, maybe kids of friends that are younger than 10 years old, they very likely will still be around in 2100. So it is not that far away. But we do know that for sure, let me say it differently, we may have the science for 2030. I don't believe we have the science for 2050, and I definitely don't believe we have the science for 2100. That doesn't mean there is the impossibility to generate it, but it's going to require a concerted effort of all of us. Therefore, the new vision for CIMMYT is to be a global thought leader and change agent for climate resilient, sustainable, and inclusive agricultural development for a food and nutrition secure world. Those that want to know more about our strategy can download it, and we have summarized it in 3 pillars. A discovery pillar, a system dev pillar, understanding that we need a systems view and that we need big programs in countries to really bring all the science together and generate the impact with farmers that we need. And we have generated something of an experimental pillar, which is the INC pillar, which is to look at, can we work with startups? Can we generate new income-generating activities? How do we better work together with the private sector? And of course, across this goes excellence in science and innovation. And let this be an invitation for several or all of you to participate with us generating that excellence. We cannot do it on our own. We need a conducive environment supported by excellence in operation, resource mobilization, partnerships, and of course, influence and advocacy. CIMMYT's 5 top-level research and innovation objectives start from wanting to be the best partner. In our objective 2, we want to generate the new innovations. In our objective 3, we understand that we need to work for smallholder livelihood trajectories so that we are sure that that science and innovation has an impact on those trajectories of families, of farmers. Our 4th objective is that we understand that it needs to be within guiding principles. Even any solution that helps livelihood transitions or generates new research and innovation, but doesn't or contributes negatively on the agro-food systems being more inclusive, effective, and productive, and sustainable, and climate resilient is of course not acceptable. And a 5th objective is we want to influence. We need to have a stronger voice as research and innovation within the agro-food systems with decision makers, be it in the private sector, be it in the public sector. We should be, as members of this community, at the table when there's big discussions in the Pentagon going on. We should be at the table when we talk about food security, global security, and simple security at its own. Simply summarized, what our strategy wants to do, and what I'm challenging all of you with, is can we shift from resolving yesterday's problems tomorrow? We did it very effectively with COVID. We observed the problem, and we ran a science community, and very rapidly came with a solution. But it was still resolving yesterday's problem that we observed the next day, the earliest tomorrow. Can we shift to resolving tomorrow's problems right today? It is a shift of taking advantage of the 140,000 different wheat accessions in the gene bank, for example, or 28,000 different maize accessions, and simply put them together and make a new seed that is relevant today while we are breeding. Breaking news for a farmer, that seed will only arrive his farm very likely 10 years from now. With the rapid impact of climate change, we're going to be too late. We will have to project future climate scenarios, future systems, and work backwards from that. Can we mine our alleles in the gene banks? And can we make no-regret investments? Can we put some of those alleles in the seeds, knowing that they will hit those environments 10 years from now? Can we predict what's going to happen? And of course, it is very difficult to predict, especially the future. So it's probably better to look at what are no-regret investments, what are future conditions. And very likely in many of the areas, those future conditions are not a projection from the past to the future, but that future condition may be closer to a current condition in another place. Europe should invest heavily in research for Africa, because some of the temperatures and droughts that are today in those areas will be current in the future in that continent, just to give one example. It is only going to work if we maintain global collaboration. Every dot on this map is a seed pack shipped from Mexico to collaborators globally. Red, be it the public sector. Blue, be it the private sector, either maize or wheat. You can see on this table many, many envelopes. Every envelope is an envelope full of hope. It is an envelope you helped to create with a new seed, resistant to diseases, hopefully more nutritious, and hopefully climate resistant. The threats are globally, and I don't have to tell this community that transboundary pathogens and pests are spreading rapidly and appearing in new areas where they weren't expected. Just to remind some of our memories, the threat is global. Just look at yellow rust. New races found in Afghanistan, Pakistan in 2018 were picked up in Ethiopia in 2020 and Uganda in 2022, and you can see how it spread on this map. Let's take another one, stem rust. New strain, nothing to do with UG99, emerged in Ireland in 2020, in Belgium in 2021, in the UK in 2022, and that is just one example. Weed blast is an even more scary one. For years, CIMMYT has been reminding since the 80s that weed blast was an issue, but back then it was only sitting in Brazil and getting investment was very difficult. Today it is spread out over Bolivia, Paraguay, Argentina, and it hit Zambia in 2018 and Bangladesh in 2016. The threat is equally global for maize. Maize lethal necrosis has popped up in Uganda, went to Rwanda, to Congo, and to Ethiopia. And it meant a 25% reduction, up to 100% reduction in farmers' fields in those countries. If the threats are global, the solutions are local, regional, and equally global. And we all have to play our role. From the more than 200 new wheat varieties released by CIMMYT over the last 10 years, to the recent blast-resistant variety that worked not only in Latin America, but also in Bangladesh, to the second-generation MLN-resistant hybrids. The solution is global, and we can learn from what we already did. This map shows the UG99 resistant varieties, and similarly, we're pulling together through the G20 a wheat blast collaborative response, and we hope many of you will participate in replicating that UG99 response model. The solution is global, but from large-scale, localized pathogen surveillance network up to the regional efforts, and I know you had a very nice presentation this morning, for example, of a West African network of labs that is being built. How can we maintain that infrastructure? How can we connect that to the global community and actually have all those surveillance networks well set up? And does it matter? It does. If we just look at this advisory system that was set up from the field through multiple collaborations, you see the logos down there, and was bringing the early warning systems together, supported Ethiopia, Nepal, and Bangladesh at the same time, reached and monitored 100,000 farmers, but sent out messages and warning systems to many, many more. It had a direct impact in how the system in Ethiopia was affected. In 2010, we had no early warning system. Extreme high risk of a rust season. We lost, due to yellow rust epidemic, 600,000 hectares were affected, not completely lost, but affected. And the estimated losses were over $250 million. In 2021, there was an early warning system operational. The same extreme high-risk conditions were there. Ethiopia, luckily, had a record wheat production. You can be proud of what you did. Unfortunately, that record high wheat production level comes with a new threat. Fusarium head blight is coming in East Africa, and urgent action is needed to tackle this increased threat. Decision makers will go out there from East African countries, apologies for that, including Ethiopia, of course pushing wheat production locally because of the impacts of the Russia-Ukraine supply. And they're not thinking necessarily about those second-generation impacts. We need an international effort to keep working on the existing surveillance networks, but especially to incorporate Fusarium head blight and other second-generation impacts. Wheat Blast has already responded, and BRAPA set up in collaboration with CIMMYT a Wheat Blast early warning system. But what is even more hopeful is that that same system was rapidly replicated in Zambia when the disease was found there. But I wasn't here to talk about how successful we already were today. It is still resolving yesterday's problems tomorrow. We all need to challenge and take that next step. Can we resolve tomorrow's problems today? Can we generate together the future of disease surveillance that is not based on past events, projecting them forwards and avoiding more of it to happen, but that actually has inherent systems that can use some of the future potential conditions, that can use the wealth of data that is available, the models, and I'm sure my colleague in the next talk will talk a bit about the power that sits in some of those efforts. And can we shift those surveillance systems from more predictive capability? And of course, that will mean monitoring and data management strategies that go throughout. And it is impossible to maintain those data systems if they're only for one objectives. Those data systems need to be multiple, they need to be connected, and of course, they need to be ethically deployed, while at the same time serve multiple purposes, so it is financially viable to actually put those systems out there. It will require multiple collaborations with different actors, from insurance companies all the way to the researchers. It will also have to be connected with how countries are measured. Can countries be held responsible to their progress to what's going to be the next sustainable development goals through those same data systems that help us from preventing some of the disasters we see on the horizon? Of course, the most difficult thing to get resources for is the prevention of something that didn't happen. So even there, we will have to work with ministers of finance to say, don't finance what happened, finance what you can prevent from happening. Tapping the vast pool of genetic diversity in wheat wild relatives, we are just seeing the first step of that. And those that are breeders here, I know you don't like those wild relatives. It's difficult to incorporate. It is messy. It is noisy. But if we can use better data, if we can use better technologies, we can make this acceptable. And colleagues from Nottingham University, from the John Innes Center, and multiple others, and the next speaker will probably maybe reference some of that work, know that it is doable. Also, how do we convince the use of, for example, more of that genetic resources equally like we did in maize lethal necrosis, but taking it the next step to genome editing, to be more deliberate in putting some of those characteristics in there. Today, we have genome-edited crops for improved food security by editing for maize lethal necrosis resistance. Once again, can we make those seeds that will only hit 10 years from now the farmers' fields by some of those technologies?
1 minute.
That will require MLN screening facilities at multiple locations, not only for the public sector, but let's share with those private sector actors. Even more challenging, it will require plant health innovation platforms where we together co-construct not only the solutions, but start from the future questions. Inherently, in any science you do, you will have to think about the scaling later on. Just saying, I do upside science and it will just reach the objective is not going to be good enough. You have to put it in a theory of change. You have to put it in an impact pathway. doesn't mean you have to do the theory of change or to do the impact pathway. You need to get better at what you're already good at, but put it in the full context and make sure you have the partners around it. It does require gender and social inclusion considerations from the design of some of the research. And especially, it means training and preparing more and better future resources. Human resources are our most valuable asset in the war against hunger. We invite you to be part of a global plant health consortium that just started, and I hope many of you will contribute. We'll prepare and we'll be a member of it, formally or informally. I want to thank our many donors, but before I close off this lecture, I actually want to read Borlaug's job description for Glenn Anderson. He said, this type of individual must not only be a top scientist, but also a doer. He, and I'm going to paraphrase this, he or she must be able to act as a catalyst, stimulate young scientists. He or she must have great physical stamina, and he or she must not accept defeat. He or she must fight back when things look dark. There's a great wealth of scientific talent in India. There's a great wealth of scientific talent in the world, in Africa, South Asia, Latin America, the Americas, Europe, that can all be brought to bear upon the research problems and wheat production problems if the right coordinator is found in this function who can bring about this chain reaction. I want you to look at the person left from you. Just look at him or her for a moment. Now look at the person to the right from you. This is not only good for your physical condition. I hope you also have observed the huge potential of leadership that is sitting in this auditorium. But as Borlaug also said, we have not fed one farmer family with potential. I want to invite you all to turn your potential in real impact. Thank you very much.
So we have time for one question only. So if you want to ask a question, please come. There's a microphone here on the back. But I don't. Or put the question on the chat.
There's one.
Yeah.
Wonderful talk. Wonderful talk. Your talk about global surveillance and scientific communication. Do we have international policy of management? Is there any policy enforcement? If not, what is your imagination?
Right. To the best of my knowledge, there is at this point no policy framework for those kind of systems. For sure, there is no policy framework for the kind of systems that we need in the future. Now, my vision is actually that those kind of policy frameworks should start from the impacts we're trying to provoke, being it those that we're trying to avoid and those that we're trying to want to happen. So yes, there's elements of ethics in that. And those of you that took science classes, I hope you also had an ethics in science class during your university. And I think those are going to become ever more important in dealing with some of those issues. But that should not help or scare us away from doing the right thing. So we need those philosophers and those that think about ethics very close to any discussion about actually our science per se. So if you don't have a good friend ethics professor close to you while you're trying to do the next big thing, find one and bring him or her closer to you to develop those elements. When you advocate with policymakers for such a policy framework, they are looking at you, your data, but turn that data into information, turn that information into decision-making, and help to turn decision-making into decision-taking. And on those steps, ethics is involved.
Thank you.
So it is my honor to announce the next speaker, Professor Fiona DOOHAN from University College Dublin in Dublin, Ireland. She's an invited speaker to speak to us about very successful research in sustainable system to increase resilience and resistance of our cereal crops. So Fiona, I let you the floor. And this is, yeah, so it works the best.
Thanks very much, Giuseppe. And thanks to the organisers and the ICPP for the invitation to speak to you today. So I want to talk to you a little bit, following on from our first speaker, about the beauty and the complexity in wheat diseases. A very recent article eloquently showed that showed the folly of depending on 3 areas in the world for the majority of our wheat supplies. And it outlined the looming crisis that there is with regard to food security, wheat being a very important source of human calories. I want to talk to you a little bit about the Global Wheat Initiative, which was endorsed by the G20 to coordinate global wheat research, to really try to bring together researchers across the world to expedite our ability to control wheat diseases, to deal with drought, to deal with stress, to enhance, to enhance food security. And in particular, there was an article that we published last year talking about the challenges for wheat production and the priorities for strategic research. And I particularly appeal to anyone involved in wheat research to get involved in the expert working group on pathogens, which actually met here yesterday as a satellite workshop, and also involved in the Wheat Initiative Crop Health Alliance, which has just been launched to develop a global disease monitoring, diagnostic and monitoring system, global diagnostic and monitoring system. So back to the topic of the talk, the beauty and complexity of wheat diseases. Anyone who's worked with wheat diseases knows that they're very complex. You have your straightforward biotrophs, such as the rusts, Fusarium head blight, which I've worked a long time on, which is a hemibiotrough. First a biotrough, then a necrotroph, and then you have things such as Septoria, which are difficult to work with, which has definitely got a necrotrophic phase, but the jury's out on what the first phase of the disease is. And these diseases have caused huge risks and cause huge challenges to food security globally. In an article, it was outlined that 80% or more of global wheat area is at risk from 4 fungi. Collectively, we'll collect the rust together, the stripe rust, the leaf rust, and the stem rust, and from Fusarium head blight. And in addition to that, approximately half the wheat area is climate suitable for Septoria triticide blotch disease. So I'm just going to outline, you've heard about Fusarium already today in the first lecture, a very important disease globally. You've heard about how there are new epidemics of this disease. It affects yield. The big thing is it contaminates the grain with toxins, mycotoxins that are harmful to human and animal health. And fungicide control is inconsistent at best. Over the last couple of decades, I've worked a lot on one of the mycotoxins, deoxynivalenol, which in addition to being harmful to humans and animals, it's also harmful to plants. It's phytotoxic. It's a virulence factor. Take away DON from the fungus and it causes less disease. It enhances disease spread in wheat. Another disease I'm going to come back to today is Septoria triticide blotch, caused by Septoria triticide. It's a very devastating disease in moist climates, and believe it or not, where I come from, Ireland, it's quite moist. We have the honour of a lot of new fungicide-resistant isolates emerging in Ireland over the recent years, and host resistance really is the only alternative to control this disease. So the cost of wheat diseases, it's estimated they're responsible for up to 20% of yield losses per annum. The rust causing up to 5 billion global annual losses, Septoria up to 1.2 billion in Europe alone, most of that due to fungicide application, and Fusarium head blight costing millions to USA growers. So back to the title, the beauty, they're beautiful, they're complex, and the complexity of wheat diseases. And I'm really actually not talking about the disease here, I'm talking about the host response. So I've worked a lot over the last 2 decades on how wheat responds to diseases and looking at the evolution of the host pathogen interaction. Why is that important for food security? We really only understand a drop in the ocean as regards how wheat responds to diseases. And I want to give you a couple of examples of that. I'm going to talk a bit about our work on Fusarium head blight, We did a lot of work a couple of decades ago on looking at how the transcriptome of wheat varieties that are resistant to fusarium and resistant to the deleterious effects of DON, how they respond to it at the transcriptome level versus susceptible varieties and populations derived from them. This one gene kept popping up again and again and again. We kept saying, ignore it, it has no motives, there's nothing in it, I don't understand it, I can't work with it, but it kept coming up. So we said, okay, let's look at it. And it turns out it's an orphan gene, a recently evolved gene, and we started to study it, and this led us to new pathways and also to look at genomic hotspots and how they respond to fusarium in wheat. So the gene is TA frog. My French postdoc who discovered this gene wasn't really in favour of the title of the gene. But anyway, Triticum ovesticum fusarium-resistant orphan gene. So Alex did a lot of work on this. He showed that this existed within the Poidia. There were no homologs whatsoever outside of that family, none in maize, rice, etc. It's a nuclear protein. We overexpressed it in Arabidopsis and shows that it was located in the nucleus. And it's responsive to fusarium and to DON production by fusarium. This graph is showing you in black columns that when you apply fusarium to wheat heads, you get optimal expression of this gene at 2 days post-inoculation. The higher the column, the more expression. We also treated heads with a mutant, mutated in the ability to produce toxin, knocked out the key gene involved in toxin biosynthesis. We showed that that still infected the plant. It was about half as infected, but there was no expression of this gene. So this was truly a toxin-responsive gene. And the dotted line there just shows we were also analysing how does the fungus produce toxin. This is analysing the expression of a fungal toxin biosynthetic gene. And the peak in that coincided with the peak in the expression of our gene. So what does that mean? An orphan gene in wheat evolved to respond to DON. This gene does not respond to anything else. We went on to overexpress it, collaborating with NIAB in the UK, and show that it enhanced resistance in overexpression line, overexpressor 1 and 2 there, at about 25 to 30% compared to the control. We then wanted to know, okay, so this tells us, well, let's step back. This tells us orphan genes are important. When you do any study in wheat, you throw away about 60% of the genes because you don't know what they do. And out of that, about 30% are probably some sort of orphan to varying degrees, taxonomically restricted. We said, well, can we discover more about how this works? So there were no motives, nothing like that. So we used protein-protein interaction studies to figure out what proteins does our orphan interact with. And one of the key proteins that we validated that it interacts with is a SNRK protein, a central sugar stress signaling molecule, AMP kinase in mammalian systems. And we showed that when we overexpressed that in wheat and when we silenced it, it did contribute to DON resistance. So this is virus-induced gene silencing, treated with a MOC or with DON, either with the empty virus, which is BSMV00, or 2 silencing constructs. And we got enhanced DON-induced bleaching when we silenced the gene. We also got a reduction in SNRK kinase activity when we silenced the gene via VIGS. So this validated that SNRK interacts with frog, and it also enhances resistance to deoxynivalenol and to fusarium. And it was very gratifying to see then that a study released a couple of years ago showed that a fungal effector, an effector from fusarium, competes with TA frog to bind and destabilize SNRK. Another frog interactor, a NAC. What's NAC? NAC are a family of plant-specific transcription factors, very, very important in biotic and abiotic stress tolerance. And we found that a very divergent NAC, or a sort of evolutionary divergent NAC, interacted with frog. It also enhanced resistance to DON and to fusarium. So here again, another gene, another interactor of frog enhances DON resistance. So what you have here is 4 overexpression lines, OE1 to 4. They have less disease. The higher the bar, the more disease. They have about 20 to 30% less disease than the wild type, which is the black bar. And then we went on to study the effect of DON and frog on the transcriptome of wheats. And actually one of the very interesting things, it ties into yield. And many of the genes that we've studied, this is just a subsection of the genes we've studied with fusarium, many of them tie into yield, which is not surprising since it is a disease of flowering heads. But so let's stop here and think. So here's an orphan gene. There's loads of these in the wheat genome. We're only tipping the barrel, and we now have all the tools to do that much faster than we did it over the last 20 years. We need to know what they do and how they contribute to resistance. for quantitative resistance. Yes, we have major genes which are really, really important, but we also need some of the minor genes. We need to understand how they work, and we need to look at genomic diversity panels in cereals to valorize allelic versions of these that might be even more beneficial. But then we started to say, well, where is frog in the wheat genome? You know, what's sitting beside it? And that's when we realized, oh, it's not alone. It's surrounded by other fusarium-responsive genes. So we started to screen the whole wheat genome for fusarium-responsive genes using a sort of a sliding window approach. And we identified what we call genomic hotspots, regions of the wheat genome, the A, B, D genome, that are enriched in fusarium-responsive genes. And we identified 44 of those regions. I'm just going to talk about a few. The TA frog cluster. It has LEC receptor kinase, orphan genes, TA frog, and right beside it there's another one which actually looks like it is the evolutionary parent of TA frog. A momelactin-like gene cluster. There's a similar cluster in rice, and these are very important antimicrobial gene clusters. And a GST cluster, which actually co-locates within a very narrow QTL. And we're now looking at this to see, are these indeed the genes underpinning the QTL? So, orphans, any taxonomically restricted gene is potentially very valuable. It's co-evolved with the plant pathogen, so it's very valuable to look at those in terms of enhancing disease resistance in current commercial cultivars, in wild relatives, etc. What about other diseases? Well, I mentioned Septoria tritici blotch caused by ZT, or Zymus septoria tritici. We've done a lot of work on that over the last 15 years. We've also identified that there are taxonomically responsive genes that interact with Septoria, some very taxonomically restricted, some less restricted. And we also show that one of those, for example, interacts with a effector from Zymus septoria tritici. So don't worry too much about this vision image, just to know that there was much more pecnidia on the leaves when we silenced the gene as compared to when we didn't, the ligase. So this told us that this orphan gene contributes to Septoria resistance. All very well. What about the real world? Well, now what we're doing a lot is integrating genomics into varietal selection. And we're doing that through two major projects. One of them is an EU project called INNOVAR, where we have 14 sites across Europe and North Africa conducting standardized VCU trials on 230 wheat varieties. And we're looking at the effect of reduced inputs, pesticide inputs, on the performance of those varieties across those sites. And we're also integrating genomics and phenomics into the varietal assessment criteria, or assessing the potential for those tools to enhance varietal selection. We've done a lot on STB and FHB. We've selected varieties that are very resistant in all sites and varieties that are susceptible. We've selected varieties that are resistant under glasshouse conditions and those that are susceptible under glasshouse conditions. And we're currently doing transcriptome analysis on all those varieties to identify genes and integrate that into the GWAS to identify genes associated with resistance to these diseases. We're now, having learned all of that, we're really stepping back and looking at the bigger picture. So an awful lot of effort goes into major gene resistance, and that's right. It's right to do that. But I'm really interested in what are the broad spectrum resistances that underpin disease resistance by biotrophs, necrotrophs, hemibiotrophs, etc. So to do that, we're looking at all the data that people have published over the last few years. Can't look at all the studies. Some of them lack some of the key data you need underpinning it. But we're taking 53 projects. We're doing cluster analysis, and we're doing network analysis. And we're really looking at transcription factors and disease resistance pathways associated with biotrophs and necrotrophs and across the pathogen spectrum. So I think that this more in-depth study of the wheat genome and of diverse wheat panels really has a lot to offer in terms of sustainability of wheat production in the future. But it's not going to do it on its own, and at the same time we do an awful lot on looking at biostimulants and the microbiome and how they might enhance wheat productivity. We work a lot on endophytes. For those of you who were here in the previous section, you'll hear that they're organisms that live part of their life inside plants, and we know that they can now help biotic and abiotic stress tolerance. We isolated probably about 2,000 or 3,000 at this stage endophytes from wild relatives of cereals from very diverse locations, including islands off the west of Ireland, which really haven't seen commercial cereal production, or fungicides or fertilizer inputs. And we've tested them for their ability to enhance abiotic stress, but also to control diseases. And we found endophytes, for example, this is just a range of them, that can reduce the severity of, for example, Fusarium head blight disease. Now we're interested in integrating that beyond that, asking, can we breed wheat for a healthy wheat microbiome that suppresses plant diseases? And we're looking at that in many projects, not least in a new project called Wheat Security, which is an EU project where we're really looking at the relationship between the microbiome and chemical inputs, fertilizers and pesticides, and disease levels, and linking that into genome and transcriptome data across the EU. So to come back to some conclusions there, what have I learned after 25 years of research? Well, I wouldn't start here, okay? And I would look at systems. And everything I do now is actually in a systems-based approach, not farm to fork, soil to human health. So all the research we do on wheat, and a lot on oats as well in Ireland, really looks at that. How does the impact of disease resistance breeding affect the nutritional quality of the end product? And it does, believe it or not. And we have to really look to the past to inform the future and valorise biodiversity, whether it's crop biodiversity or environmental biodiversity, and integrate all these tools into our crop breeding programs and into our biocontrol programs, and really integrate it into much more broader diversity panels to be grown around the world. So I'm just going to finish off by thanking people in the laboratory and collaborators, and I'll take any questions.
Thanks, Fiona, for the very nice talk. And we start to think about how to prepare for tomorrow with resilience and resistance. Is there any question from the public that you want to put in the chat? So there's one, it's very practical. So how to apply endophytes in the field in the future or present wheat production?
So we've done a lot of work on endophytes, and to be honest, it's very, very good for seed. We apply it as a seed dressing, and we also do foliar treatments. Both have varying efficacy. I would say seed treatments are very good. They're very good for seed-borne diseases. Particularly, I'm thinking about barley, actually more than wheat, and Romularia and net blotch. But we also apply them as foliar. And diseases, we haven't had success to date, at least, with diseases, many foliar diseases, because there's such a big window of opportunity for the pathogen. We've had great success with Fusarium because there's a very narrow window of opportunity. So it really depends. So I would put a lot of effort into Fusarium and endophytes and seed-borne diseases and endophytes, and also looking at endophytes that not only benefit disease. So we're also looking at drought and nutrient use efficiency. And we have some key stars that really perform across the board. And that's the ones we're looking at, and variants of them.
Thank you. So now we receive a lot of questions. I think we'll forward to you later. But do you want to select another one? Yeah. So there's a lot of questions about biocontrol and endophytes.
Not that one.
Yeah. So
This one, did you find any tricotine? Oh. Fiona, did you find any tricotine glucoside transferase?
Did I find any tricosine glucosyl transferases? So these are genes that can glucosylate deoxynivalenol and render it non-toxic to the plant. Yes, we did, but we never worked on them very much. The same reason we didn't work on UGT glycosyl transferases, because in your belly, in your stomach, you can cleave off those molecules. So we really sort of veered away from molecules or genes that might modify a molecule in a way that might not be stably modified. So yes, they're there, but we didn't really work too much on them. That's not saying they're not important, and there could be ones that are very stable, but I've just steered away from that.
the last question is what is the impact of legislation and in general policy on developing biocontrol agent?
Yeah, so that's a whole new area, and it's an area that we're doing a lot of work with various companies in terms of registering the products, and rightly so. It has an impact. It depends very much on where you get them and how they function, etc. What's the impact? It's huge in terms of costs, and you really need to make sure that you really have stars. So we start off really looking for organisms that have very good biostimulant potential drought potential in that, and then also test them for biocontrol efficacy. We'll still have to go through legislation for using them, or comply to legislation for using them for that purpose, but that's not our first goal. Okay, cool, thank you.
Thank you very much. And I can see this is a very resilient audience. And I also notice you are becoming a little bit food insecure. I'm Lise KORSTEN from the University of Pretoria. I'm also the co-director of the Centre of Excellence Food Security. I'm also our society's chair of the Food Security Commission. And I hope that afterwards you're all going to come and join us and make a difference. You've heard the challenge. We have to put ourselves in the middle to really find tomorrow's solutions today. We've heard some excellent presentations, and I'm now going to move over to our last speaker for this afternoon. And let me introduce Dan, and I'm just quickly getting his presentation on board. And Dan, he's the chief scientist of Oak Ridge Laboratories in the USA. We've heard network analysis. He's going to expand and going to tell us about opportunities with AI. We are moving into the future, and we are looking forward to your presentation.
Thanks, Lisa. Thanks for the opportunity to talk today. When you invited me to give this talk, you told me to stretch the boundaries a bit and be more holistic in a One Health framework, so you may get more than you bargained for. Adventures in systems biology and sort of a One Health framework here. So we're really interested in tackling lots of different problems. And as a systems biologist, we're interested in a broad range of systems, but really looking globally that as a global ecosystem, we're all connected in one way or another. And we as humans in the newly crowned Anthropocene age are perturbing that environment. And that environment is now returning the favor. COVID-19 may well be an example of the environment returning the favor, as is climate change and various things we need to deal with. So we're studying effects of changes in climate that I'll touch on today, the need for not just carbon neutral, but actually carbon negative societies to really reverse the trends that we're seeing, which brings plants right into the equation with carbon sequestration. Of course, sustainable agriculture and energy. And I'll touch a little bit on the precision medicine efforts and, in fact, other feedback loops of future pandemics that we'd like to prevent. So our view of the world really is on a whole range of different species, plant species, microbial species, and then biomedical systems, whether they're humans or biomedical model organisms like mice and Drosophila. Out of all of that, you have lots and lots of omics data now becoming more and more available, lots of phenotypic information, lots of environmental information, and we need to combine and integrate all that in meaningful ways. So we typically use whole ensembles of different types of algorithms, be they AI, explainable AI, data analytic, you name it, we're probably using it somehow or on our exascale computing platforms now at Oak Ridge. And why exascale? Well, biology is really, really complex. The epistatic potential of alleles combining in a single genome, plant or human, is on the order of 10 to the 180. Okay? For context, there are about 10 to the 27 stars in the universe, maybe 10 to the 83 atoms. So I'd like to tease our astrophysical colleagues, if you want complexity, look at biology, right? If you're looking at a big scary problem, we shouldn't say, that's an astronomical problem. We should say, oh my God, that's a biological problem. We have incredibly complex systems that we as a community need to study and understand that combinatorial complexity in meaningful ways, and that faces us every day. So we're using these combinations of approaches to try to capture these patterns coming out of lots of different types of omics data, often at population scale. And we tend to capture those patterns as
multiplex networks representing those relationships as a mathematical representation of a complex system. And now we also have a structural biology arm of my lab led by Erica Prates that converts that into the three-dimensional interactome. And the AI components that feed that, things like AlphaFold 2 have become quite popular tools that are allowing us to do new things. All of that really is to get down towards conceptual models, actionable experimental conceptual models, things that design the next experiment to validate these conclusions, to do selections for genome editing and breeding, to bring new things to the fold. So I talk about omics. We now call this the OM. If you name an omic technology, we're probably using it somehow, ranging from genome to epigenome up the biological hierarchy to phenome. And we're really using these tools to try to capture patterns within and now across all of those omic layers in interesting ways that really starts to understand mechanism in new ways. So on the plant side, again, sticking to this view of the world, the sweeping data through algorithms, going towards networks and trying to get to mechanism. One of the workhorses in our bioenergy programs is poplar trees, black cottonwood. They're very fast-growing trees, so they're native to the Pacific Northwest of North America. So colleagues over the past 10 or 15 years have collected a diversity population. This is an undomesticated species, so no historical breeding bottlenecks. So collecting across the native range, across a lot of environmental clines, to build populations and now have been deployed in many, many different common gardens, ranging from British Columbia all the way down to Sedona, Arizona for extreme environmental shifts. All told, I think something on the order of 20 to 30,000 trees have been deployed in these common gardens over the years. That gives us a tremendous resource for a range of things. Among them, the microbiome. So we've actually done lots of different campaigns across these populations in different environments, both OTU-based as well as sequence-based. And what I'll talk about today actually is a data mining experiment on data that was originally targeted at EQTN, so trying to find, do GWAS on expression values across the population, but collected expression data from about 500 or 600 different individuals of the population in multiple tissues. And we use KMER-based approaches to mine the cryptic microbiome that's hanging out in all that plant transcriptome. When you're sequencing plant tissue, you're also sequencing all the transcripts from the microbes living in that tissue. And it turns out anywhere from 5 to sometimes 15 or 20% of the sequence space is actually occupied by microbes in that tissue. So we've built KMER profiles of every available genome on the planet, not subsamples, but every single one we can get our hands on. So we can do fairly high resolution KMER mapping to identify the taxa that are present across this population. We find thousands and thousands and thousands of taxa in these different tissues. Anybody who's looked at microbiome data knows it's very gnarly. 0 is an evil mathematical number. It has sparsity problems. So we actually built AI pre-processing pipelines to separate the portions of the microbiome that have reasonable statistical properties that are useful for GWAS. Then we took every taxa resulting from that pipeline and used it as a phenotype for GWAS in the population. We noticed among them that 2 different species, a Streptomyces and a Pantoia, so as the previous speaker talked about, the microbiome is really important for both biotic and abiotic stress resistance. So we're really interested in understanding what are the host genes that are controlling colonization of these different microbes in different tissues. We noticed these 2 species both map to both of the inositol transporters in the host genome, in the poplar genome, both the vacuole transporter as well as the cross-membrane external transporter. So we said, well, inositol is a sugar, and it's also an internal signaling molecule in plants. It's part of inositol 6-phosphate signaling cascade. So maybe we're hitting a receptor and we're seeing the signal cascade. So we knocked out, actually transitioned to Arabidopsis and ordered up the knockout lines for pretty much everything in the inositol signal cascade. Made no difference whatsoever on colonization. But the knockout of those 2 transporters in Arabidopsis recapitulated the phenotype, had a big impact on colonization. So we said, well, it's also a sugar, so maybe the bacteria are catabolizing that. So we knocked out the catabolic enzymes in the bacteria, made no difference whatsoever. And the we here is these validation experiments are happening in Sarah Lieb's lab, first at the University of Tennessee, now she's at Michigan State University. And the next clever step that they did was actually a motility assay of putting inositol in the media and measuring how fast these bacteria swim in a halo assay. And lo and behold, that's the bacterial phenotype, is that when it's not catabolism at all, it's not a sugar source, it's not internal plant signaling, it's an inter-cross-kingdom signaling molecule that the plant is broadcasting by pumping inositol out in the environment, specific bacteria, come here, colonize me, I'm over here, come this way. And so this is a new function for inositol that's never been known before, just came out in Communications Biology last month. But the power of mining this sorts of data, finding that cryptic biome, and then using the power of GWAS to figure out what's going on the host side and starting to get hints of what's happening on the bacterial side. So we have this new conceptual model of this cross-kingdom interaction that inositol is now part of. Now, we also want to understand the microbial interactions, and traditionally we haven't been able to use tools like GWAS in microbes because there's no sexual recombination. Every daughter cell is a clone of the parental cell, so you have no genetic resolution. Everything is NLD. Well, in collaboration with Josh Michener's lab at Oak Ridge, we figured out a way to actually have bacteria recombine and select for those progeny that are recombinants, that are shuffling the genomes of two very different parental strains. And in selecting for that, effectively you're selecting for the equivalent of bacterial sex. And now you have recombinant resulting genomes that have shuffled the genomes, and they become a GWAS population. And we've built tools to deal with some of the peculiarities of microbial genomes to be able to do GWAS on them. And now then you need to do high throughput phenotyping. So we've built phenobots for rapidly screening these fairly large bacterial populations for different phenotypes. This one's looking for visual phenotypes that you can do for things like motility assays or biofilms. We're coming up with hundreds of different phenotypes we can now screen bacterial populations for. And in this case, we, in fact, automated a motility assay. So did a motility assay across an entire shuffled population and are able to do GWAS and find a very nice peak here, starting to nail down what are the important genes involved in motility itself for bacteria. So we're now able to do GWAS both on the host organism as well on the microbes. We're also starting to do this same sort of approach for pathogens. Septora, for example, we're doing in the Septoria muciva, which is a canker-causing pathogen in poplar. We're doing GWAS on the Septoria population and figuring out how to combine that with the host population. Again, your combinatorics go up when you have thousands of individuals on both sides of that, but we're figuring out hopefully some clever mathematical approaches that we can do two-way GWAS at the same time and really understand the interaction between host and pathogen mechanisms simultaneously. Now, we've done this across the entire microbiome, so we have lots and lots of associations, and we want to start to integrate those multiple members of the microbiome together. And so running through these network-based approaches, we can move towards conceptual models integrating the results of multiple associations at the same time. And this is starting to help us tease apart what's been a long-standing question for us is, in any host genome, you have a limited number of receptors you can put on your cell surface. Let's say that there's 1,000 receptors that you're going to put on your cell surface. But the number of microbes you're going to be exposed to in the surrounding microbiome, and soil microbiomes are incredibly complex. You can't always have a one-to-one relationship between receptors and members of the microbiome. And what we think we're seeing is the cell's doing a very clever thing. It's deploying an array of detectors, and it's using that array to do combinatorial signal processing. more than one receptor will have different fidelity for the same microbes and will also work across multiple microbes. And so it's receiving a complex set of signals from the cell surface, and downstream with the intersection between these signaling pathways, it's doing combinatorial signal decomplexing, really. Combinatorial signal processing to understand what is friend or foe out there. And so, okay, if you have 1,000 receptors and you just use them in pairs, well, 1,000 squared is 1,000,000. All of a sudden, you can handle that sort of combinatorial complexity just with pairs, and we're seeing it uses higher combinations than just pairwise. So the cell is actually doing very sophisticated mathematics in sorting out what are the members of a microbiome community that are trying to colonize it and how it wants to respond, who to let in and who to try to fight off. We're also, of course, very, very interested in how all phenotypes, including whether it's friendly microbes or pathogens, how responses are conditional on the surrounding environment, how abiotic stress may be affecting phenotypes, be they physical or pathogenic. So we put together lots and lots of different data types, many of them that are capturing seasonality across the time of year, because of course your environment, unless you live at the equator, your environment is constantly changing over the course of the year. So instead, and this is one example of aridity, and you can see the distribution and the values are shifting as you progress across the entire year. So instead of doing one association against the genome to climate type as a proxy for phenotype, we're actually doing it for every day of the year. So you're actually generating a GWAS, a Manhattan plot, for every day of the year over a time series. So we're calling this a genome-wide association of time series, or GWAS. And we can see when we do this that the time series actually matters. So this phenotype is actually aridity, so how humid or dry an environment is. And here we're seeing a SNP and a gene involved in wax formation, the cuticle of the leaves, so important for controlling water loss. You see the association in very specific times of the year, starting in mid to late summer. The rest of the year, we don't see it at all. So if we just took the average of this, we probably would have missed the association. So again, the time series matter. If we take a different climate type, in this case wind speed, we see a SNP involved in a gene involved in leaf formation, so when your tender little leaves are coming out in the spring. Sensitivity to wind stress is important. So there's an evolutionary adaptation in the spring, but the same climate type associates at a completely different time of year in the winter for a cell wall-related gene probably involved in trunk rigidity. So for how likely you are to be blown over in a strong wind. So different adaptation in a different gene for a different function in a different part of the plant that, again, we would have missed if we tried to average it all together. So we're doing this across the board for lots of variables and, of course, creating more network layers of climate type to gene associations that we can feed into our network associations and then see how those are intersecting with pathogen phenotypes, with microbiome phenotypes, and with the physical phenotypes to get, again, this complete systems biology view of the system. Now, if we're able to now start to understand the genetic architectures responsible for abiotic stress, and we want to breed or genome edit our way towards more resilient phenotypes, as both previous speakers talked about, well, we need to know what are the possible regions around the world that have specific environments, that have specific climate types that we can optimize for. And as was pointed out in the first talk, today, some of today's environments may be in Africa or South America, may be tomorrow's environments are Europe or North America. And so understanding the relationships we currently have and seeing how they're changing over time and being able to project into the future allows us to design genotypes, cultivars that we can test in current environments and future-proofing future environments. So we want to find what are those climate type zones. So we do, with these data sets, we have environmental vectors for every GPS coordinate on the planet, every point of dry land on the planet, except Antarctica, because we're not planning on growing plants anytime soon in Antarctica, and if we are, we're even in deeper trouble than we think. And we do all against all. We build vectors and do all against all comparisons of all of those, both two-way comparisons and three-way comparisons. So n squared and n cubed style comparisons. Save the significant relationships between them as a network. And once we have a network representing the environmental relationships around the planet, we can use our network tools. One of the tools is network clustering, in this case, Markov clustering. We don't just do this once. We do this also across a sliding window over the past 65 years of real-world data that we have, so we can see how things are changing over time. Two-way and three-way network topologies are somewhat different, and we'll see the effects of that in a second. When we cluster this network from the two-way comparisons, we see some of these broad biogeographic patterns we would expect to see, but we also see we're lacking some resolution, like most of the eastern half of the US shows up in one cluster. While we know there's a lot of environmental variance in the eastern US, but when we look at the three-way networks, the clusters are much closer to what we think the biogeographic resolution should be, and we have ways of internally validating these, some of them being mountain ranges. When you are moving laterally through a mountain range, your altitude is shifting very rapidly, so you have very common shifts in microniches in mountain ranges, so you would expect to see extreme fragmentation, and that's exactly what we see in the Himalayas, in the Sierra Nevadas, in the Andes. We can actually pick up mountain ranges by the environmental fracturing. So with that on board, we think that and other species distributions we're looking at give us some more and more confidence that these climate type zones are now reasonable representations of these environmental relationships across the world. As I mentioned, we're also doing this in sliding windows, so we can see how this changed. So now we have 65 different networks representing environmental relationships around the world, and we can compare then every point of land on the planet in its relationships of what they used to be to what they are becoming, and use that as a measure of relativistic change. How are environmental relationships, which are important for habitats and ecosystems, how are those changing over time? When we animate those changes, this is what we see. The blue color here is a relatively stable environment. Environmental relationships are fairly consistent over time. The more orange you are, the more you are changing. If you're worried about pandemics, there are a whole range of hotspots here in Eastern Australia, Indonesia, Bangladesh, India that are lighting up. If you're worried about things like Ebola in Central Africa, they're lighting up. If you're worried about Zika in South America, they're lighting up. If you're worried about agricultural systems in these areas, these are big areas of concern. If you're worried about carbon, you start looking at the Arctic. Over 33% of the world's carbon is locked up in frozen peat bogs under the permafrost in the Arctic. 33% of the world's recalcitrant carbon. As that permafrost is thawing and those ecosystems are re-emerging, we've been up sampling in the Abisko Research Station in northern Sweden, and the microbiomes that are emerging are enriched for methanogens. So we're not just converting that recalcitrant carbon into CO2, we're increasingly converting that into methane. Why does that matter? Well, methane is a 20-fold stronger greenhouse gas than CO2. So the weather we're experiencing when you walk out the door and it hits you in the face, not what you expect in Lyon. The risk, of course, is we're accelerating this cycle. So that's sort of a happy Monday, we're all screwed message. But we can do things about this. And part of this is, in fact, in Plant One Health. Plants are a major opportunity to suck more carbon out of the air, sequester it in the ground, and start to reverse this cycle. Pulling off these calculations I just described, we hit 1.37 exaflops of performance. That's the fastest calculation done in human history. It's a nearly 170 zeta-op calculation. That's one of the largest calculations done in human history. About 10 to the 17th edges in the network we originally calculated. We think that's probably the largest network calculated. So now we know how microbes are controlled, what affects abiotic stress, where things can grow, what are the different regions. We want to do more phenotyping to integrate the picture. We've just are burning in a new phenotyping facility at Oak Ridge that has multiple hyperspectral and non-hyperspectral imaging modalities. This facility alone can generate 3.5 petabytes of data a year. We decided we needed to come up with a metric for that, so we're calling that an F-ton of data. You can fill in the F however you would like. The PC version would be a flip ton of data. But we're busily developing new algorithms then to deal with those layers of data and the hyperspectral nature of them, because now you're not dealing with RGB images, you're dealing with data cubes and polytopes that we need to integrate through. We're also doing a lot of field-based phenotyping data, and I want to run you through a leaf campaign that we've been doing the past year or so. So why leaves and why vein architectures of leaves? Well, if you're trying to suck carbon out of the air and put it into food or bioenergy or into the ground for carbon sequestration, leaves are where it's happening. Of course, for every molecule of CO2 that you're fixing into a sugar, you're using 6 molecules of water. That water has to come up through the vascular system from the roots through the stem and out through all the veins in the leaves to get the photosynthetic tissue. All that sugar you're creating via photosynthesis has to get back out into the rest of the plant for biomass, food, root systems, bioenergy. So key linchpin of the system. There's tremendous variance in leaf architectures and vein architectures in the same species. These are 2 different leaves from the same population. You can see by eye they're quite different. You can see by distribution of veinicity they're quite different across the population. I wish my postdocs worked this fast, but this is a time-lapse video of a team in the field scanning leaves. We've actually built parallelized leaf scanning now, 10 parallel scanners running at once. We've scanned over 20,000 images of leaves in the past year, and that number is increasing. But once you have the images, getting that vein architecture out is a non-trivial problem. To trace by hand the vein architecture takes a postdoc basically a full day to trace 1 leaf. When you start talking about thousands, the initial study had about 3,000 images. We figured that was 5 person-years of work it would take to trace this out by hand. So that would probably lead to suicidal postdocs. And so not a good outcome, and we don't want to wait 5 years. So we have a new AI approach that solves some of the challenges we often face in plant phenotyping. And traditional AI imaging approaches are very data hungry. They often need hundreds of thousands or millions of examples for training. They need lots of annotation. Well, what do we have in leaf scans? We have no annotation other than what we can trace by hand, and we have a few thousand images. So the clever idea here was a method called few-shot learning, which is with those few. Initially we used 5, eventually we used 8 leaves, so a week and a half of work for annotating the vein architectures. You can break that down into little 3x3 pixel tiles. And with that, you gain about 10 million. You get about 10 million samples you can train on, and that actually works very well then to use this region-growing few-shot learning approach to capture the entire vein architecture that actually outperforms humans. Once we did the validation of this, the AI approach was finding veins the original postdocs missed. With that, then, that gives you whole ranges of phenotypes you can extract from this. All told, we extracted 100 new leaf phenotypes from this single data set, validated them, and are doing GWAS in a range of ways on them. But now that we have 100 phenotypes, we're also looking for those phenotypic relationships. So we're using a different AI approach, an explainable AI approach, to find the relationships, the predictive relationships between phenotypes, and they break out into nice biological units. And here we're going to zoom in on vein and petiole thickness phenotypes, with a few phenotypes that are quite connected and some that are quite disconnected. Doing GWAS on all these, GWAS has its own challenges with false positives and false negatives, both due to type 1 and type 2 error balancing, but also because simply of gene assignment. Most GWAS SNPs are between genes. You assign it to both genes on both sides of that SNP, and by default, you know that half of your assignments are going to be wrong. So networks to the rescue on the hypothesis that genes that work for common phenotypic functions are going to have tight network connectivity to one another. We actually use the networks in a way to represent extent biology, filter and categorize. So we actually can take, in this case, from several phenotypes, there's about 386 genes, and build a dendrogram with all the false positives in there, but then run it through a different multiplex network algorithm that actually looks for that connectivity within each of those clades in the dendrogram and filters out the false positives, only retains those
Sorry,
that have...
Daniel. Can you go to the conclusion slide? Because we'd like to ask some questions to connect the other talks.
This gets us towards new ways to look at multiple phenotypes at the same time to get towards conceptual models, and we're really going to blaze for a lot of slides now. There's a biomedical side of this that shows that we know how to get to a conceptual model. With the same approach, in this case, neuropsychological conditions, we can use this method with functional partitioning to drive us towards the conceptual model that really falls out of those network associations. Gives us new drug opportunities, et cetera. Jumping really quickly through pandemics, now our conceptual model has to incorporate even broader sets of data. And what we thought was a 10-year AI goal to build predictive models of the next pandemics. And these pandemics have mortality rates on the order of 75% when they hit humans. Okay, COVID, really terrible. We've lost millions of people, sub 1% lethality. If these paramyxoviruses evolve aerosol-based transmission, we are done, right? There will be no time to develop vaccines or counter therapies, and the politics we've seen in COVID, if the virus doesn't get you, your neighbor will. So that's another Happy Monday message. We're all screwed. We don't want to react to the next pandemic. We want to prevent the next pandemic with this sort of lethality. So we're actually building AI models that can predict likely zoonotic spillover events in these flying fox bat populations, and narrows this tremendous environmental complexity down to three variables. And it turns out these variables are once again associated with plants, because what do flying foxes feed on? Eucalyptus flowers. So what we're finding are the key abiotic stressors that lead to a zoonotic spillover is actually plant biology. If you screw up plant biology and stop flowering, the bats go into stress, shed a lot more virus, leads to spillover events. Pretty good accuracy models. We're submitting the paper any day now of what we thought was a 10-year goal. 1 year in, we actually have models that are working. Lots more to do, but we can start to solve these sorts of problems. We're crossing a lot of scales. We actually go from quantum level information up to planetary scale calculations, able to do this sort of work with the extreme compute power we have at Oak Ridge, lots of different funding sources, and wonderful teams that have been involved in so many different components of this work around the world. And this phenomenal team of students and postdocs and staff I'm so fortunate to lead at Oak Ridge. We have lots of educational and sabbatical and collaborative programs, so please collaborate, come and see us. And with that, we could take questions. Sorry, I'm a little over time.
Thank you very much. Thank you very much, Dan. That was wonderful. You've closed the loop. I'm not going to allow questions because we've just gone over our time, but you can respond on the questions in your own time. We started this morning with Cindy and One Health, and we most certainly had challenges both from Cindy coming all the way through the presentations, and Bram, you made the message very clear. To all the plant pathologists, we have to put ourselves in the middle of One Health, and now you also know very clearly we have to be part of the solution, which we've now heard can be done today for food security for all. So with that, I want to close the session. I like the precision health. I thought that was brilliant. And we need precision plant pathology to solve the future's problems. And with that, thank you very much for your time. Please do come tomorrow. I just want to say we have 2 more food security sessions, one tomorrow at 10 o'clock. Then we'll go from talking to doing, and we'll hear how people are solving our food secure problems. And then Thursday at 6 o'clock, we will have another food security session. Thank you very much.